So this brings us to our last lecture and back to, in a way, human evolution. Our speaker is the distinguished non-David Prize laureate, Mark Feldman. And if I may quote from the decision of the Prize Committee, Professor Feldman has produced conceptual results of broad interest in the domain of animal and plant evolution. His work has led to highly focused insights on cultural significance, such as out-of-Africa model of human evolution, as well as cultural preference in different civilizations. His work not only explores basic scientific topics, but investigates the societal consequence and of the conclusions he draws from in terms of models of evolution. Professor Feldman originated the quantitative theory of genetic modifiers of recombination, mutation, and dispersal. His work was the first to show that the pattern of interaction among genes determines whether sex will evolve. And this is just a fraction of the superlative that Mark Feldman deserves. So he's going to talk about, in a way, human evolution. And his title is The Canadian Fur Trade and the Evolution of Tuberculosis. Please.
First, I want to begin by thanking Avram and Arnon for organizing this symposium, and to point out that this is actually the 41st anniversary of my first collaborative paper with a professor from Tel Aviv University. And since that time, I've had a huge number of collaborations with professors and students. Ilan doesn't even remember that it is 41 years since we published our first paper. So it's been quite a while that I've had a close connection with Tel Aviv University, and it continues. I'm going to talk a little bit today about something related to humans, but the actual topic is about bacteria and the interaction between bacteria and humans. How do we, Avram, how do we move it on? Is this forward?
Yeah, I think so.
I'm going to be talking today about tuberculosis. And what you have on the screen right now is a map of the frequency of tuberculosis per 100,000 population around the world today. This is there has been no update on this since the 2004 WHO figures, and you can see that there are a lot of places in the world, here, here, and here, where you have up to 3,000 cases of tuberculosis per 100,000 people. 1/3 of the world tests positive for tuberculosis, and there are between 2 and 3 million deaths a year due to tuberculosis. So TB is a very serious problem. It is also caused by a bacterium called Mycobacterium tuberculosis. The bacterium itself is not easy to study, and the reason is that it doesn't culture well in the laboratory, to the extent that we don't even know what its division time is under laboratory conditions or in the lungs, when it is most of the time quiescent. That means it's not causing disease. A couple of years ago, Ruti Hirschberg, an Israeli postdoc, and my student Misha Lipatov published this tree, which is based on the complete DNA sequence of 70,000 bases of the bacterium itself. Now remember, I'm going to be talking mostly about the bacterium. And these 70,000 nucleotides were chosen because they were supposed to be genes that had something to do with resistance to drugs for TB, none of which are very successful. And we formed a tree based on the number of single nucleotide differences, and these single nucleotide differences, which are called SNPs, single nucleotide polymorphisms. You'll hear me using that term SNP throughout the talk. And what you can see is that there is a tree where the European and American clade, the India and East Africa and East Asian clade, two West African clades, one here, and this one is from the Philippines and the rim of the Indian Ocean. But in addition, we sequenced the TB complex. Now, this is not called Mycobacterium TB, it's called the MTB complex. And these are from animals. And this one here is Mycobacterium bovis, which is from cows, and originally, for a long time, was thought to be the origin of tuberculosis in humans. That has been disproved now, just fairly recently. But these are other animals that were sampled in Africa and elsewhere. And up here, you see a very strange animal, which is only found in Djibouti. It's called Mycobacterium caneti, and it is extremely variable. It has 100 times more variability in it than any other TB strain, and nobody knows why. It's called Mycobacterium canetii, and it really forms an outgroup, and that's why this line here tells you that it's a lot different from these other ones. Now, this is a picture painted by Frances Ann Hopkins. It's actually in the archives in Canada. You can see it in the museum in Ottawa, actually. And it's a canoe full of Canadian French fur traders. And she painted it 200 years ago. And it's very strange because she painted herself into this picture. There were no female fur traders, but she thought it was a nice thing to be able to do. And this is an Indigenous person who is helping the traders as they went across Canada to trade furs with the Indigenous people in the far west. I'm going to talk a little bit about the background, about the migration dynamics of the bacterium itself, and about the TB epidemic database. And I'll just tell you now that we've recently completely sequenced, that is all 4 million bases of about 50 strains of TB, and we found some very strange things going on that are actually about to be published. Things like a lot of lateral gene transfer, for example. That was not expected. This is TB bacterium, this thing here, and it's attached to a macrophage. And actually, there is a gene which facilitates this attachment, which is called the toll-like receptor. And the toll-like receptor, TLR, is actually one of the most polymorphic genes in humans. And there's good suggestions from our own work on Africans that toll-like receptor is under strong selection. It's a gene that's been under strong selection in humans. This recent review says that if we can understand pathogen evolutionary and phylogenetic patterns, then we can understand the effects of infectious diseases on human history and therefore on human evolution. And that's one of the most interesting things to do, is to try to figure out how the pathogens that humans faced over the last 100,000 years, since Tabun or Hafsa or Schul, what they did to our genomes. these are lineages that we've studied, and I've told you that we have one way of looking at genetic variation in the bacterium, and that is by simply looking which nucleotides vary. There are two other ways to look at the variation. Mycobacterium TB is subject to an insertion sequence called IS6110. And this is a little piece of DNA that jumps around in the DNA of the bacterium, and we can characterize each continent by the exact pattern of the IS6110 insertions. And that's called a fingerprint. And we showed about 6 years ago, in collaboration with a very good professor of infectious disease at Stanford, that every continent was characterized by a different pattern of insertion sequences or fingerprints. And unfortunately, that professor left to head up the Gates Foundation's TB program with a budget of $800 million a year, so I could understand him leaving the university. Unfortunately for us, we had a 2-year hiatus, but then a very, very good young professor of infectious diseases came along and wanted to collaborate with us, joined our group in population genetics, and the stories that I'm going to tell you now have been the result of collaborations with Caitlin Pepperell, Dr. Caitlin Pepperell, who's in the infectious disease division in general medicine at Stanford, and my excellent graduate student, Julie Granker. These strains here are based on the fingerprints that I told you about, and they're different lineages. And you can see that the American lineage, European lineage, even reaches to Australia. And then you have several African lineages, these different colored ones here, lineage 5 and 6, that are not found anywhere else in the world. And those in India and the Southeast Asia are not found in the Americas. So as I said, continental division of the bacterium itself is obvious. So we have these different ways of making the trees. These are based on SNPs. These are based on the fingerprints that I just told you. And these are based on the third type of DNA, which is called deletions. And in many bacterium, when you look and compare the bacterium, you see whole blocks that are missing. And those are called deletions. And you will see a very important deletion, which is going to play an important role in what I'm going to tell you. So this is the beginning of the story. This is from a book by Ferguson in 1928, which tells the story of TB epidemics. And I actually blanked out the tribes' names here of the Indigenous peoples of Canada for ethical reasons. And what you see here is that in the early 1880s, they had a fraction, this is per thousand, at least 13% of the Indians, of the indigenous Americans, got TB. Why? This was exactly the time when they were placed on reservations. Up until this time, they had been essentially in the nomadic area. And just before this happened, around about 1870, was the killing off of the buffalo herds. And they would have trains with people standing at the window shooting the buffalo. So there was no more food left for the people of Saskatchewan and Manitoba and Alberta. And the Canadian government decided the only way to save them was to put them on reservations. When they put them on reservations, the children were educated in very tiny rooms with no ventilation. The result was this terrible epidemic of TB, which lasted about 10 years and killed off about 14% of the indigenous people. There were two other epidemics, one around the turn of the century and another one in 1920, that actually plays a big role in Saskatchewan. Now, where would the bacterium in TB, where would it have come from? How did it get into the indigenous peoples in the first place? They didn't have it before white people came to the New World. There's one hypothesis is that it was there anyway, just like Helicobacter pylori, which is the bacterium which causes stomach ulcers. And if you look at the pattern of variation in Helicobacter pylori, it's exactly the same pattern as you see in the genes of humans. And it has what we call the serial founder effect. As you leave Africa and move eastward, you find less and less variation in that bacterium all the way through to the Americas. And the reason why it compares with humans is that it's one of those bacteria that recombines freely. There aren't very many such bacteria that we know recombine freely. And Helicobacter pylori is one of them. So the story of the evolution of Helicobacter pylori could be the reason that the indigenous people of Canada got it, that it came when the original Canadians came, which is probably between 15 and 30,000 years before now. Another possible reason is this huge migration boom that was apparently coincident with the epidemic. In this period between 1871 and 1911, a huge migration came into Canada. And between 1900 and 1917, another huge migration. This is the number of people that actually migrated. And where did they come from? They came from all of these places here. They were a mixture of Euro-American and Asian peoples who decided that Canada was a good place to live. Now, the lineages of the frequencies of the bacterium in 4 Canadian indigenous populations, these are the indigenous populations, and this is the French population of Quebec. Now, the French population of Quebec is extremely interesting for human geneticists. And the reason is that after the fur trappers came in the 17th and 18th century, there were no female French-speaking people, and the King of France gave a ransom to women who decided to go to be the wives of the trappers in Canada. So about 1,000 women took up the offer and moved to French Canada, and they are the founders of French Canada. And if you look at the diseases of French Canada today, you will see the level of strange diseases that are due to the fact that this is a very small founding population. But the point that I'm making here is that this one here is a deletion of the bacterium, a piece that's missing that's found in Quebec. This is a strain which is standard European everywhere, and it's the lab strain which everybody does their studies on. And this Quebec strain is found about half of the people in Saskatchewan, more than half, 62% of the indigenous people of Saskatchewan are carrying this deletion, which is found in Quebec and in Alberta and in Ontario, and these are all indigenous peoples that I'm talking about here. How did the Quebec strain form? Actually, it's a recombinant between this strain here and another strain which is called RV 1765, and the deletion that we're talking about is this, that includes this deletion of this fingerprint piece here. This insertion sequence is gone, and the Quebec strain is easy to recognize in the lab as having this big piece of DNA that's missing, and it's a Quebec-based strain. Now, if we look in the Saskatchewan indigenous peoples, and we just take that DS6 Quebec, which is that deletion strain, then these people here, all of these people, and these are the size of these circles is the number of people, and these are different regions that we have in the Saskatchewan Indigenous peoples, you find that there are some from this region here, region 4, which is this yellow, and this one here is region 2. You have larger numbers, and sometimes you have very small populations, which are just 1 or 2 people. These lineages over here are unrelated. They're part of the ones that are not the Quebec strain. But all of these are in the Saskatchewan Indigenous peoples, the Native Americans of Saskatchewan. Now here's a map which shows the routes that the fur traders took. And there are 2 routes that I want you to focus on. One is this one, the red route, and that was all the trappers in the Hudson Bay Company. They're British. And the blue is the Northwest Company, this one here, which is the French company. And they went all the way through here, through Saskatchewan, all the way into British Columbia. And they crossed over from Saskatchewan into Alberta, and actually went all the way up into the Northwest Territories by boat. And this map will show you also some things that we'll see later, that the DS6 Quebec is half of the people in Quebec carry that strain, and look how frequent it is in the Indigenous peoples of Alberta and Saskatchewan. Now, I don't want to go into too much of the mathematics that we did here, but the history of this prediction is the first French immigrants come in 1608 to Canada, and the fur trade starts in Quebec around 1760. Actually, there were traders in small numbers at 1730, but this is where it was institutionalized, and the fur trade expanded to the west in these dates between 1730 and 1870. And in Alberta, it started about 1750 and finished in 1870. These are different estimates of the dates at which we use the DNA of the bacterium to estimate how long ago the bacterium was founded. That population of bacteria was founded. And that involves a lot of computer simulation, and what you get are very big confidence intervals, and part of that is because of the small samples, the relatively small samples that we have. But the estimate that we get as the median of our estimates of 1797 is pretty much plumb in the middle of the historical prediction. Remember, these estimates plus their credibility intervals are taken by using the DNA and using a mutation rate which we don't know from TB, but we know from another bacterium called Yersinia, and another bacterium. So it's not yet known what the mutation rate in TB actually is. It'd be great to do that, and we think that by the next few weeks we'll have a very good answer for that question, because we'll have complete sequences on the 50 strains that we've been looking at. So what this tells you is that the fur trade expansion appears to be coincident with the historical predictions from the literature of the migration caused by the 5,000 people who became fur traders from French Quebec. Now what you see here is called a minimum spanning tree, and this minimum spanning tree takes the data from all of these 4 areas, Quebec, Ontario, Saskatchewan, and Alberta, and it connects them by using a 4th kind of DNA, which is called mini-satellites. Mini-satellites are strings of DNA that are 10 to 60 bases long, and they reside in about 1,000 places on the TB bacterium. and they're polymorphic. They vary from place to place, and that means that we can form a minimum spanning tree like this. And the characteristic of this that I want you to take away is that it's characteristic of a demographic expansion. This is the kind of tree that you would see if there was a demographic expansion of the bacterium. It's not like some other trees that you'll see, like this one. You'll see there's a cluster here, a cluster here, a cluster here, and a cluster here. These are other strains of bacterium. But I'm going back too far. No, I'm going the wrong way. I want to go backwards.
You can just scroll around.
I'm trying.
Go out this. You want to go back?
Yeah, back. That one, yeah.
Okay, you just scroll here.
Oh, okay. When we ask how much migration actually happened, then there are a lot of different ways you can do the estimate of genetic migrations of the bacterium. And it depends on which statistic you use. But the actual analysis of molecular variation, which we're using, this number here says that about 8% of the strains came in from outside, and they came from Quebec. So this is the pooled what we call pooled lineage migration rate, and that is a good framework for estimating the migration rate. Now, when you ask how many people migrated, it's much more complicated than how many bacterium migrated. And the number of people who migrated, the population size is N, M is the migration rate, and MTB migrates with these people, and unfortunately, every different statistic gives you a different number. This is just how it happens in, when you're doing analysis of molecular variation, that you find that you get different results. The historical says 5,000 people moved. The analysis of molecular variation using one technique says that this was the number of population size times migration rate, and another technique, 3.62. So there's no way around that. It just is a fact that different statistics will give you different estimates of the migration numbers. Now this, I show you this map again because it's an example of what we call the serial founder effect. Now, Cavalli-Sforza and I invented this term to explain why it is that when you move from Africa to the Americas, you get less and less genetic variation. And the idea is that the first group that left Africa 60 to 70,000 years ago, probably from East Africa, formed a colony, and then the next group was a subgroup of that group, and then the next group was a subgroup of that group. So you continued the effect of genetic drift, getting more and more strong as you moved across the Bering Sea and into the Americas. And the fact of the matter is that the least genetic variation today is in Indigenous Americans. They show the lowest amount of heterozygosity and the greatest length of their DNA without any polymorphisms at all. And that's the serial founder effect. Now, if you look across here, you'll see that Quebec has the most variation. Just look at the different numbers. And then over here, you see in the indigenous populations of Saskatchewan and Alberta, you see the least variation. So it's happening in the bacterium as well as it's happening in the humans. Now, this pattern actually is an interesting one because it shows the number of distinct haplotypes based on restriction fragment length polymorphisms, and it's contradictory to what we expected. Because if you remember the map, you saw that Saskatchewan was actually further away, so, came before Alberta. So we would expect these two to be reversed, these two curves to be reversed. And we asked, why is that? And the answer came up in a very interesting way. There are two parts to Saskatchewan. One is the part in grey, and one is the part in white. And the part in grey is what we call remote Saskatchewan, and that retained a barter economy with no financing, and we call it remote Saskatchewan. These parts here were already trading with the French for money. And when you just look at the remote Saskatchewan people, you find that this is the amount of variation in the remote Saskatchewan, almost none, and everywhere else you see some variation in the bacterium, including non-remote. So the remote Saskatchewan, non-remote Saskatchewan has incredibly different biology because the French people were able to do different kinds of trading out there. Now, when the French came, they actually stayed there. And there were records of the French traders settling in the West, staying there 6 to 10 years, marrying with the indigenous peoples, and the only way that TB can be transmitted is through droplets from the mouth, and it's most likely that these French Canadians who were in the fur trade transmitted the Quebec strain to these indigenous peoples. And the other evidence for that is that you do not see any of the British strains in that area. Why? The British formed homesteads. They were not fur traders. And they had a rigorously enforced social system where none of them married into the indigenous communities. So that explains why we don't see any of the British Canadian bacterium in the indigenous American communities. Now this is a lot of mathematics here, and what we tried to do was to figure out whether the timing of 1930 would make a big difference to what we saw in the number of strains of bacterium. And the reason 1930 is a big day is because that was the first day that airplanes started to land in remote Saskatchewan. And these were what were called bush planes, the little planes that transported the people instead of going by boat. Going by boat was hazardous. It took a huge amount of time. And from this point on, people came in droves to Saskatchewan, and they came by planes. There were whole companies devoted to transporting people to Saskatchewan by bush planes. And this is an estimate of what is the most likely ratio of the effective number of strains in 1930 to the effective number of strains now. And you can see that it's about 0.08 is about the effective number. And that means that at 1930, there were probably 2.3 strains of the bacterium in remote Saskatchewan, but in the whole of Quebec, there were about 18 different strains in that year. So the Saskatchewan people had formed a bottleneck, a small bottleneck of the bacteria. So the conclusion that we came to is that the TB that we see in Saskatchewan is actually due to the dispersal via the fur trade, that it didn't exactly happen with the epidemics, but the epidemics were the result of social structures that were imposed on the indigenous people by the Canadian government. we still see persistence of small populations, isolates of Mycobacterium TB in those populations. There was a marked bottleneck, 2.3 strains versus 18, which is a marked bottleneck, a sharp reduction in the number of strains. And there's no evidence of transmission from the homesteaders. So the next steps that we are going through is we're collecting data on about 80 Aboriginal communities in Canada, and we have good evidence from the literature, because the Canadians kept very good data on the frequency of TB and the dynamics of TB. They kept good data on the demographics of both the Indigenous populations and the non-Indigenous populations in the same area. And one of the things that we really want to do is to go back and sequence the genomes of the Indigenous American community from the same area, especially to look for those areas which we know, like NRAMP1, that are genes that are associated with resistance to TB. And that's a big project, but we're about to begin just the human part. This part is finished, and it'll be published within the next few weeks. Thank you.
Thank you so much, Mark. Any questions? So let me conclude our symposium. I think that those last lectures presented here really unequivocally show Dobzhansky's statement about the importance of understanding evolutionary theory in biological research at any level of organization, from bacteria to human. To my opinion, this is the beauty of evolutionary theory. It explains genetics, have led to the discovery of epigenetics, explain animal, including human behavior, and finally population structure and population demography and epidemiology. So I hope that you all join me in this conviction that whenever we study biology, we should study it in the light of evolution. So I would like to thank everybody for coming, especially the speakers for speaking here so nicely, and thank you all.